Okay, hello everyone. Thank you so much for joining again in our e-seminar series today. It's it's our honor to have Professor Sean Wu with us from Stanford Medical School, University School of Medicine, with us, who talks about the stem cells at the crossroads of cardiac development and tissue engineering. But before that, as always, I go through some housekeeping notes. You can ask your question in the Ask a Question box on the bottom of your screen. And then there is a chat box that you can chat and socialize with other people. And you can send your greetings from wherever you are in the world. So we are happy to see that you're joining us from different parts of the world. So by that, then our next speaker is Professor Todd Orr from Department of Chemical Engineering, McMaster University. Basically, he's talking about hydrogels in tissue engineering, biomaterials. If you want to get more updated information about the upcoming seminar, just we have a Twitter account with the hashtag TransLBME. So please follow that account to get the upcoming seminar schedule and updates. And last but not least, we would like to also thank our sponsor who provided the platform and the support for this e-seminar series, Montreal Transmedtech Institute, for their supports. By that, I introduce our speaker today. It's our great pleasure to have Professor Sean Wu. He's a clinician scientist, is a board-certified cardiologist who specializes in treating men and women with cardiac diseases such as coronary artery disease, cardiac valve disorder, rhythm disorder, cardiac oncology, cancer drug toxicity, and cardiac preventive management. His research career has been dedicated to making fundamental discoveries in developmental and regenerative biology of the cardiovascular system and apply these findings to prevent and treat the damaged heart. He has made a major contribution in the isolation and characterization of cardiac progenitor cells and their differentiated progenies from mouse embryos and human iPSC. He has considerable expertise in human induced pluripotent stem cell modeling of cardiovascular diseases using genome editing technology and tissue engineering approaches. His recent work focused on single-cell transcriptomes of the developing cardiac conduction system and bioinformatic annotation of single-cell RNA sequencing data. His work has been funded by the NIH Directors New Innovator Award, NIH Directors Pioneer Award, the California Institute of Regenerative Medicine, the NHLBI Progenitor CellBiology Consortium, and is currently supported by NIGMS R01 multidisciplinary collaborative grant and multiple collaborative NIH R01 grant and Established Investigator Award from American Heart Associations, among others. So this list goes on and on. By that, it's our pleasure to have Professor Sean Wu with us, and I invite him to share his screen with us and start his presentation. Thank you so much.
All right. Well, Hooman, thank you so much for that kind introduction. It's a real pleasure to be here, and thanks a lot, Mohsen, for inviting me to do the seminar. And I'm very excited to see how this technology is taking on to really deliver science in a much bigger way, as you guys are doing. And definitely look forward to have a nice dialogue with everyone here in the audience about the research work that we've been doing. So let me just check. You can see the share screen. Is that right? Okay, so I'm going to switch to presenter mode since I now, after that, can't see anybody at that point. So I guess if you see something weird, please let me know, since I'm just going to be talking to the slide and not sure exactly what else is going on. Yeah. Okay. All right. So as Huma mentioned, I'm going to be talking about the intersect between heart development and tissue engineering, and I always start my talk with lab members that we have, since they are the one that's doing all the hard work, and I really want to first acknowledge for all the time that they spend, really working hard at addressing important biological questions. So the research work that my lab has been doing, and the ones that I'm going to focus on is the intersection between cardiac developmental biology, which is where my lab had first started when we were in Boston at Mass General Hospital, and connect that with cardiac regeneration tissue engineering, which has been the work that I started sort of returning to when I went to Stanford, where I was originally was an undergraduate student in mechanical engineering, and that's sort of how I had reconnected with that when I moved my lab from Boston to Stanford. I'll sort of touch on a little bit of this reprogramming and disease modeling, since the cell that I'm going to be referring to a lot is the iPS cell, which is generated from reprogramming, and iPS has certainly been known as a really great in vitro models of various diseases. So I'm going to touch a little bit on that. Just a little bit overview of the different projects in heart development. You can see heart cardiomyocyte conduction, cell differentiation, and using single cell RNA-seq approaches to delineate these cells. So that's been a major focus. In cardiac regeneration tissue engineering, we've been generating various iPS line coming with various reporters for labeling the cardiac subpopulations such as atrial or ventricular, and more recently the left side and right side of the heart for RV, LV as well in iPS cells, and trying to take the cell population into some aspect of a 3D construction, such as using 3D bioprinting strategy. And from the reprogramming and disease modeling aspect, we've been using iPS cells for disease modeling and drug screening, and also some direct reprogramming strategy, converting cardiomyocytes into other cell types as well. So this is kind of in the nutshell what the various work that has been going on in our lab for the last 10 to 15 years. And here are some of the publications that we have published in these different areas over the last 10, 15 years as well. So, where I will start is, the way that we've been thinking about what tissue engineering should look like in the context of how the heart is normally made, which occurs, as we all know, in heart development. And so from the side of heart development, you normally have this very small embryo structure, the blastocyst, that generates the 3 germ layer, the gastrula, and that goes through a process of morphogenesis and heart development to generate the looping stages of the heart. And then through further expansion of the different chambers, eventually give rise to this mature functioning heart. And I think the mature functioning heart has been the goal that many people have been trying to achieve from different ways. And tissue engineering certainly has been one of the major areas that people have been trying to do this with. From tissue engineering, there have been many different cell types that people have been trying to use. But I think in recent years, the iPS cell, particularly human iPS cells, have become the dominant cell type of use in tissue engineering, mainly because the versatility of these cells can expand indefinitely to as many cells as you need. And then with the right recipe, you can differentiate them into various cell type of interest. And then so these different cells, for example, the cardiac cells, are then put into various different 3D engineering formats into an assembly of structures that hopefully resembles what a mature functional heart is. So I think in a sort of a very conceptual view, these two different fields are interested in the same thing, but of course, comes from it very differently. And we've been, working at the heart development for a long time, and we've been applying some of the principles of heart development in tissue engineering. And, so we're hoping, that someday, we can really bridge these two different fields into a developmenteering, so that's my own term, that essentially allow us to not just trying to create engineered tissue as, starting from bricks and mortar kind of way, but really more creating engineered tissue by growing and developing them in a sort of controlled and defined fashion with engineering technology. So that, is the hope, the aspiration for what developmenteering, would do. And then, so the, the iPS cell, that I'll get into more detail, that I think most people, know by this point, is a reprogramming event that starts with the somatic cell from the patient or from any individual by generating these pluripotent cells and taking the cells into differentiation processes. You can then use these purified cells that are differentiated to do many different types of applications, such as disease modeling or drug screening or toxicity assays, and also taking the cell back to the same person, the patient, for cell-based therapy, ultimately as a way to, have a renewable cells to treat disease organs. That I think, is a process that has taken quite a long time for many different reasons. But some of the successes more recently has been mostly in disease modeling. And then, so I'll sort of touch on that just a little bit in some of the work that we've been doing. And so Arun Sharma, who is now a junior faculty at Cedars-Sinai, but used to be a graduate student with both Joe Wu and I, had published a paper using iPS-derived cardiac muscle cells as a model for a disease entity called viral myocarditis. And I think, especially given recently with COVID, the infection of viruses into the heart generating myocarditis, I think has really taken on, quite an interesting new direction. But the idea back then when we started out was to essentially use iPS to generate cardiomyocyte, which normally you can't get from human, to be able to plate into the dish. But in this case, he was able to take the iPS-derived cardiomyocyte and treat with a surrogate of a cardiotropic virus, the Coxsackie B3 virus. And when the virus has been tagged with the luciferase for visualization, he was able to then show that the viruses is able to grow and expand in the cardiomyocyte, similar to the way the viruses would normally attack the patients. And here in the picture that you can see with progressively increase in time when the viruses are incubated, with the cardiomyocyte, the cells start to, develop cytopathic effects, and then eventually beating stops and detach to indicate that the cells have essentially died. And so the development of the iPS as a viral myocarditis model was something that we started back, almost 8 years ago, and now many people have used this cell as the SARS-CoV-2 viral platform to study the pathogenic responses of the cardiomyocyte to the SARS-CoV-2 virus as well. And another line of work that we've been doing related to other disease models. So some of the papers that we publish in disease models are listed below. But since my lab study heart development, we've also been interested in trying to use iPS to address a hypoplastic left heart syndrome. And the hypoplastic left heart syndrome, you can see here in the diagram. On the left-hand side, your normal heart has 4 chambers. In the hypoplastic left heart syndrome, the left ventricle, so the main pumping chamber on this right lower corner, is small and somewhat shrunken. And this small chamber occurs due to a variety of different reasons, but most dominantly has been thought of as being a obstruction issue in the inflow mitral valve, so the valve from the left atria coming down to the left ventricle, and also valves in the aortic outflow going out to the aorta. So the obstructioning of the flow in these valves was thought to be the mechanism for the disease pathogenesis. So what has been described is the no flow, no grow hypothesis for the onset of hypoplastic left heart syndrome. But because many congenital heart diseases have a genetic etiology, those usually ends up causing some intrinsic defect within the cell, such as the cardiomyocyte. We wonder whether or not there could be an intrinsic myocyte defect for the patients with hypoplastic left heart syndrome. And then so with the iPS cells, we were then able to start exploring, are there issues like, do the hypoplast patient make less cardiac myocyte, so less ability to generate cardiomyocyte overall? And we don't really see that when we measure the iPS differentiation by the percent of the cardiac cell as measured from troponin T expression. But when we then look to see whether or not that there is a increase or decrease in their ventricular cardiomyocytes, since the hypoplast left heart syndrome affects the left ventricle, we did notice that the making of the ventricular cardiomyocyte, as indicated by the expression of a gene called MYL2, myosin light chain 2, which is ventricular specific, there seems to be somewhat of a lower percentages than we normally get for the ventricular cell, which is mostly in the 80-90%. So that raises some sort of suspicion. Maybe the cardiomyocyte formation to the left ventricle is blunted. But it also then makes us wonder, does this indicate whether the myocytes themselves are functionally deficient as well? So we perform an assay using the pattern single cell contraction that was pioneered from Beth Pruitt. And what we did is we were putting the control iPSC from healthy individual, as well as the hypoplast individual iPS cardiomyocyte, and measure the contractile force in the single contractile assay. And you can see there's a general decrease in the contractility of the hypoplast derived cardiomyocyte. And when we looked at the transcriptome changes in the cardiomyocyte, we noted that the control versus the hypoplast are quite different. And you can see in this single cell assay, where each individual dot represents one transcriptome of a single cell. When the cells are similar to each other, they would lie in the same area. When the cell has different transcriptome, they would separate. And you can clearly see the separation between the control and the hypoplast cell. When we looked at what types of genes are differentially expressed, it was quite interesting that we noted there were decrease in expression of a lot of metabolic markers. So ATP generating processes are generally blunted, and while the sarcomere, contractile force generating muscles, were increased, probably as a compensation for some of the deficiency that the cells seem to be unable to contract at baseline. We measure the metabolic consequence using an assay called the seahorse, and this measures the oxygen consumption rate as a representative to the oxidative phosphorylation from the mitochondria. And again, you can see that the control overall had a much higher level of metabolism compared with each one of these hypoplast lines to support the deficiency in these cells to maintain their contractile respiratory, energy production state as being the cause of their deficit. So these are sort of the two examples of iPS's disease model that I want to show you from what we've been doing, both in the developmental setting and as well as in the infectious disease setting. I'll also touch on just a little bit about the iPS-related cells in cell-based therapy, since this has been the area that many people who use this iPS cell have been interested in. So the work that was initially started from Chuck Murray's lab, this was published 7 years ago, using human ESL-derived cardiomyocyte injected into non-human primates. What was shown in the study was the presence of these rather large tissue area in the histology section that represents human-derived cardiomyocyte. So there's quite a bit of promise in the possibility for this remuscularization process in the injured heart. And more recently with iPS cells, and in this case, this is a primate iPS cell injected into primate heart by Yuji Shiba, who used to be a trainee with Chuck Murray. And they've also demonstrated similarly these large area grafts in certain sections within the animal heart as an indicator for repairing heart disease. And so I think what these studies illustrate is that now there are ability to transplant and engraft cells into the heart. But one of the issues that have come up as they found out from doing this is that almost all of the animals develop lethal heart rhythm, ventricular tachycardia, while even receiving a very potent suppressant of arrhythmia, amiodarone. And the suggestion from these arrhythmia is that it's probably due to the cells themselves not really being as mature as you would like, and also probably the organizationing is rather disarray. You can see in the histology picture, they're not very organized and aligned as you normally would see in the native cardiomyocyte. And this was something that from quite a number of years ago we were addressing, so, back in 2010, that we realized these cell-based injections give you a rather disorganized and chaotic arrangement, which decreases their electrical homogeneity that you need and decreases fourth generation when they're not aligned, and which then leads to decreased therapeutic potential and increased hazards. And you would certainly like to find ways to guide and organize these cells into a much better pattern fashion in order to reduce some of the toxicity and increase their therapeutic potential by doing that. Which then brings me to sort of the engineering aspect, since organizing cell required some additional tools in order to coach the cells into the proper arrangement. So some time ago, when I was in Boston, we had a collaboration with Kip Parker's lab, and the collaboration started because of this paper that was published when Adam Feinberg, who used to be a trainee with Kip Parker many years ago, and what Adam was able to do was something quite clever. He was able to take the in that case, mouse, rat neonatal, cardiomyocyte, and organize them in these patterned fashions in order to generate a directional contraction. So in their paper, they compare between the anisotropic, which is sort of your directionally aligned, versus isotropic, which is sort of a randomly oriented. And what they're able to show that when you have this directionally aligned, so the anisotropic aligned cardiomyocyte, the directional force gives you the ability for the organized forces to generate a much sort of greater directionality, rather than the isotropic, which just randomly contract in all directions and does not exert a sort of a work activity behind it. So we collaborated with them and look at a mouse ES derived cardiomyocyte in the study that was published from my collaborator then, Ibrahim Domian in Ken Chen's lab. And in this paper, we used mouse ES derived cardiomyocyte, align them in the pattern fashion like what Kip Parker's paper that had been done, and show that these aligned cardiomyocyte, which is put under this what we call diving board, can go through each cycles of systole and diastole with contractions. And here's just a video of what this looks like. So basically, spontaneously, the cardiomyocytes are placed underneath this backing, and they contract spontaneously at a rate of 20 beats per minute. And you can then pace them at a faster rate if you like. So in this case, this is pacing at about half a hertz. And then you can increase your pacing, if you want to say 1 hertz, and now it paces faster to indicate the ability for the tissues to capture. And what this allows us to do is to then be able to measure the forces based on the deflection on a stiff substrate, how much the cardiomyocyte is able to deflect and generate force. And that allows us to then use for different force disease-based, like cardiomyopathy, for disease modeling and drug screening and toxicity assay using these engineered platforms. And so, from that, which is now quite a while back, and we, since coming to Stanford, have decided to also work on tissue patterning, but now trying to get into a larger tissue constructs. And so this was work done by Vahi Sapushin, which I think is in the audience. And Vahi is now a faculty, tenure track faculty at Emory, with his own lab, doing quite a lot of very interesting tissue engineering work. And when he was with us at Stanford, we collaborated with Uken Demirci, who is in our radiology department, who has been creating lots of engineering tool, and one of which is this bioacoustic assembly. And this bioacoustic assembly is quite interesting, which essentially, I sort of, in a very layman's term, called it putting the cells on top of this little platform. You can see this round disc with a square in the middle. Cells are putting up in this platform, and the bottom is a giant speaker. So it's as if you're playing different music through the speaker. If you want the cells to move one way, you can play rock music. And if you want them to move a different way, you can play classical music. And so essentially, by tuning the different amplitude and frequency, and this is an example where the reds are beads just to see how they're supposed to look. And you can see that the tuning of the frequency and the amplitude allows you to make the patterning of these different particles in the different shape. And these process works quite fast. If you sort of don't like the pattern that you see and you want something else, you can certainly decide to just change to a different frequency. And then so I just switched to the time when we decided to make a slightly different pattern. So you can see easily within the next few seconds, and all these beats are going to start moving because the frequency has now been changed. And that's essentially in real time how fast this whole thing has been tunable to create different pattern. So as expected, you can generate lots of different patterns of all sort with this acoustic tuning of the particles. And since these are beads, just for illustration, we've taken on and put cells into the same format, and these cells essentially gets shaken by this hydrodynamic force in these Faraday waves to then create the various patterns. And then when you cross-link in a fibrin matrix, the cells gets essentially fixated into their positions, and you can then culture these cells onto the conventional tissue culture dish in the various different patterns of interest. And so this is what it looks like histology. So at the beginning, day 0, they're just randomly organized, and then you start the process of patterning, and they start looking like the E pattern and the F pattern, where there are areas of bands and areas of gaps. And we specifically look for cell density between the random versus bands and gap, and clearly the bands has much higher cell density. And then when you look at the histologic changes, there's also lots of troponin positive stain within the band area, relatively small in the gap area. But importantly, when we measure the function of the contraction, and you can see that they're very nicely organized calcium transient that passes through the band and very consistently across multiple areas of interest, whereas the gap area or the random area does not exhibit much of this cardiac contraction activity. So that was in relatively more in the 2D format patterning, and the 3D format patterning, sort of forced us to move into 3D bioprinting. And the 3D printing is certainly an area that many tissue engineers have been working with, and us, along with everybody else, has been interested in taking the 3D printing strategies to organize a more functional 3D tissue. Some of the earlier work in 3D printing, seen here from a Korean group, shows that you can essentially, put the pattern substrate into various different nozzle, and in this case, a more sort of a extrusion-based method of 3D bioprinting, but there's certainly quite a number of other strategies that have come along in recent years. I certainly don't have time to go through and review all of them, other than just to describe what we have decided to do some years ago, collaborating with the company Advanced Solutions, where they developed this 6-axis 3D printer that allows you to essentially take different material in various different cylinder and print in a little bit more flexible, rather than just a gantry-based XYZ printing. We have a sort of printer that allows you to move in space according to whatever the sort of design software parameter that was introduced. So over the years, we've sort of explored this printing strategy to make various things, including vascularization within tissue constructs. So taking various different cells that we had interest in trying to pattern, the various inlet and outlet into engineered tissue constructs. Eventually, what we ended up is something that is a much smaller scale, because I think the limitation have generally been having enough cells to print. So this, as one example, and from what Vahi and others have been working on, is to generate a small microfluidic channel system where you have essentially a plastic button in and plastic button out, and with small channels printed in the middle, about 150 micron or so, that are made from sacrificial material, and then you can encompass these into either cell-based or PDMS-based material to be able to flow the liquid through these channel ins and out. And we sort of explore various different cells that could be printed in this format, and develop these sort of microchip-based printed circuits. For example, printing things like liver tissue into one chip and printing cardiac tissue into another chip that could be then connected in these circuit for looking at the cell and cell-cell interaction between these different organs. And so this is still quite a bit of work in progress, because one of the major challenges we still have is to create the true vascularity within these tissue construct still remain quite difficult. We would like to figure out how to do a much bigger scale, because these small chips are fine, but if you really want to make a full-size human, or even a portion of the human heart wall, you really need to, have a way to, organize these cells in a format and have enough cells along the way. And so, we decided to address the issues of having enough cell, because as we were thinking about the complexity of generating these very large engineered tissue, we realized, if you were putting into cells that are on the order of the whole organ, you need roughly 4 to 6 billion human cardiomyocytes. Even if you're only making a scaffold of a human tissue, it still requires you to have somewhere in 0.5 to 1 billion cardiomyocytes. And all these numbers are tremendously large when it comes to engineered tissue. And then so we have worked on this generation of the cardiomyocyte part, which has historically been very labor time intensive and costly, and it happens to have a lot of batch to batch variability as well. And so the solution that we came up with actually came from the work from a recent postdoc, Jan Boekema, together with the current postdoc fellow, So Lee. And so what they did in this paper that we published last year in Cell Stem Cell was to utilize a very well-known developmental signaling pathway, the Wnt, and together with some cell biomechanics in the form of cell-cell contact interaction, and found that if you simultaneously activate Wnt and reduce cell contact or prevent cells from contacting, you can synergistically induce this very massive expansion of iPS-derived cardiomyocytes. And then so I want to go through just a little bit on this, because I think this strategy really is now giving us a way to create large-scale engineered tissues. And so the Wnt signal, I think some of you probably know quite well, involves multiple different components. So the Wnt binds to the receptor frizzle, which partners with a co-receptor LRP. Here you see on the right-hand side, Wnt on, the Wnt at the top, collects all of this complex molecule together, including a major one, GSK3. The GSK3 historically is a inhibitor, and then this inhibitor prevents the beta-catenin from entering the nucleus, but upon engagement of Wnt, the GSK3 is inhibited, and allowing the beta-catenin to enter the nucleus to activate the left TCF system. And we use an inhibitor of GSK3 beta to mimic the Wnt binding for most of our experiment and induce the beta-catenin entry. And so one of the early paper that Jan Boekema did as a PhD student in Boston was to show that in the developing mouse heart, if you decrease Wnt activity, so here in this case, LOF is the loss of function of Wnt, compared with CTR, the control, there's decrease in the muscle thickness in the developing heart when you inhibit Wnt signal. So this says that the Wnt signal is required for the heart development normally. But we also notice somewhat interesting that the gain of function, meaning if you increase the Wnt signal, it doesn't increase a lot of cardiomyocyte, which was a little bit puzzling, and we try to take a look at this phenomenon in the in vitro culture system. And then so together with many other papers that have tried to do this as well as what we have, we noted that when you use Wnt stimulation of iPS in culture, you get slight increase, as with most people have, but not as dramatic of an increase as you would like, so only maybe 1.2 to 1.4 fold. And so we wonder, maybe when you're growing cardiomyocyte in the dish, after the cells have been made to proliferate, say with Wnt treating, eventually the cells will come into contact. And we know that from the cancer literature, this contact inhibition is a major impediment to cell proliferation in cancer cells. And then so we wondered whether the same phenomenon also operates in cardiomyocytes. And that sort of launched off a series of study that became part of what came into this paper, which is looking at the differentiated but immature cardiomyocyte. Here you can see when we take the iPS cell through cardiac differentiation, by day 12, we then either don't passage or passage densely, meaning put them back into a small surface area space, or we passage them sparsely by putting the cells into a much larger surface area space, but maintain the same media volume overall. And what we noted was quite interesting. Only when you grow the cells in the sparse condition will you get this very major fold increase. So this is log units on the y-axis of number of cardiomyocyte increase. And that was quite tremendous because we've never seen an expansion, quite like this with iPS cardiomyocyte in culture. And this was also confirmed by showing the increase in the proliferation marker Ki67 within the cardiomyocyte population. We then decided that this cell contact by sparse plating potentially could be combined with the Wnt activation and to see how many cells we can get eventually. And so this was one of the most interesting things that we were able to demonstrate in the paper, which is essentially just continuously passaging and treating with this GSK inhibitor CHEER and show that you can take a small number of cardiomyocyte in a small dish into 10 plus flasks of T175 full of beating cardiomyocytes, and we show that the purity of the cardiomyocyte is maintained very high with the CHEER treating, while the control without CHEER treating actually declined because of the overgrowth of the non-cardiomyocytes with each of the passaging. And we're able to show this consistently across many different iPS lines cardiomyocyte, as you see in the F panel. In general, we can easily now reach close to 1 billion cardiomyocyte with each differentiation run, which is pretty amazing because that gets us a lot closer to the number of cardiomyocyte we originally envisioned for generating whole human heart. And the degree of expansion is easily 200 to 300 fold in most of the cell line, which was pretty impressive. And then, so to demonstrate some of the sort of tissue engineering interest, we created this sort of small scale engineered tissue in these sort of 6 well plate platform, 12 well plate platform, and show that these expanded cells, which was expanded up to day 42, compared with the cells that did not expand taken to day 42, and then constructed into these engineered tissue, and followed by 25 more days of maturation. And we're able to see that despite having gone through the expansion process, the expanded tissue is still able to subsequently mature after the expansion. And when we measure the amount of force generated, they continue to maintain quite significant force, perhaps even slightly greater than the non-expanded cell, which probably gets overtaken by fibroblasts that are within the tissue somewhat. So we're pretty excited that the expanded cell have not altered their capacity to expand. And so the final thing that we have done, and I want to just show very briefly in the next 2 slides, is that we've taken the playbook from a small organism, the fly, which has this very linear heart tube, in trying to figure out, can we create a heart pump in the tubular fashion in this way? And so this is a brand new collaboration that we've been doing with Mark Schuyler Scott, who came to us from the Wyss Institute, Jennifer Lewis's lab. And he's been published some very interesting 3D bioprinting work. And this is one of the recent ones that was published in Science Advances, where they essentially done a sort of a fresh-like printing that Adam Feinberg pioneered originally, and show the creation of various vascular network within a block of tissue. And we utilize the strategy that they have to essentially create somewhat of a little medium-sized scale. So in this case, about the size of the whole rat heart of a tubular construct, including cardiomyocyte and fibroblasts, in endothelial. So in this 10:1 ratio, to try to create something that hopefully would give us that contractile tissue construct, like the way you see in the fruit fly, and perhaps to identify ways for this contractile tubular construct to be of use in the tissue engineering fashion, such as repairing the Fontan circuits and other vascular needs. So that's what I have. So in summary, what I've talked to you today is the creation of functional cardiac tissue requiring close collaboration between developmental biologists, biomaterial scientists, and biomedical engineer. I've talked about directional control of cardiomyocyte alignment and how that enables synchronized beating and productive force generation. I've also mentioned that using bioacoustic patterning of human iPS cardiomyocyte allow us to efficiently generate different aligned pattern of engineered tissue constructs. And finally, I described the use of Wnt signal in inhibiting cell-cell contact to synergistically induce the massive expansion of iPS-derived cardiomyocytes, and to use that to create full-scale cardiac tissues. And finally, I want to thank, again, the lab folks who have done the work, particularly Soa and Yan, and also Vahi on the bioacoustic patterning, and also with many of our collaborators at Stanford, and also ones that are outside of Stanford. But certainly last but not least, for all of you for listening, and also our funding agency over the years for the research work. And I would be happy to take any questions if anybody has.
Mohsen, you are mute. Mohsen.
Oh, okay. Sorry. So thank you very much, Professor Wu, for your very inspiring talk. I personally learned a lot from this presentation. A part of my research is related to tissue engineering, and then it's fascinating to see the advancement that you've made in the field of cardiac tissue engineering. So while you're taking a break, I just wanted to announce that our next speaker is Professor Todd Orr from McMaster University. Make sure that you mark your calendar to join his talk. It will be a very interesting talk on hydrogels and their applications for tissue engineering and drug delivery. Also, please ask your questions in the Ask a Question box so we can keep track of your questions and then ask them from our speaker. So with that, I'd like to start from the questions from the audience, and then we'll see how things move forward. So the next question is from Michael Barton, who was a speaker at our eSeminar series before as well. So fascinating description of aligning cells on the speaker. Extrapolating, could one map the frequency response curve of the healthy cardiac tissue portions of a specific patient and then play this to align the graft to match for this specific patient, all in preparation for implantation?
Yeah, so that's definitely a very interesting idea to explore. We had not looked at whether healthy cardiac tissue have a particular frequency response. I would say that the bioacoustic strategy and how versatile it is for tuning different frequency and amplitude could very well be tuned into something that would be able to match for patient-specific needs, and it sort of depends on whether or not that patient, have a need that we understand what would be the most optimal pattern, to match. So I would say the tool itself is definitely capable of quite a lot of things, and we haven't tried to figure out if there was a patient-specific, organization or arrangement that would be most optimal. But once we are able to know that, I think definitely the technology would be able to satisfy and generate the pattern needed for that.
Yeah, that could be a cool idea actually. If it works, it's a personalized approach to treat cardiac diseases. So the next question is from Rohin. How do you align cardiomyocytes in bioprinting?
Yeah. So in most cases, the cell alignment requires some degree of tension, which provides the directionality. So if we create the 3D constructs, it would still require some anchoring of that tissue because some years ago, I had this interesting conversation with Ninette Bursek, who I think many of you know as a cardiac tissue engineer. And what Ninette, described to me, I think illustrate this point very well, is if you take cardiomyocyte on a 2D sheet and there's not a relatively stiff backing that the cardiomyocyte are on, they will just immediately crumble like you crumble a saran wrap. So you cannot have a cardiomyocyte that sits on a completely soft substrate beneath it and still expect that to maintain its shape once you give the usual media with calcium and glucose for the cells to start contracting. And then so what that means is you basically will always need to have the cardiomyocyte either 2D or 3D anchored on the ends in some way. And those anchoring, you probably have to provide the tension on top of just the anchoring in order for the cardiomyocyte to align. We have found over the years the contracting itself does exert a little bit of alignment tension, but probably not as much as if you really go into the usual 10% length increase of a tension on the tissue, which give you much more of a exertion. And I think cardiac cell likes having what we call preload. Like it likes to be under tension loaded for it to function. It doesn't like to be completely soft, with no load, just intrinsically as the way that cardiac, muscles are. And so, I think the alignment that if you want is going to have to be built into the engineered tissue by your own creation of tension in the direction that you want the muscle to be aligned.
I see. And do you think that maybe when you print the size of the fibers, the diameter of the fibers, or including nanoparticles within the bioink could help this directional controlling or directional control over the cells?
Yeah. There's been, interesting work that people have in creating like these micro grooves and then have the cells put onto these micro grooves as a way to control. I would say that's somewhat similar to what, the Kit Parker collaboration that we had before of putting, specific cell adherent substrate like, fibronectin laminin, and then alternate the strips with pluronic that are cell repelling. And so you have the cells organized in that way. But these are all mostly 2D strategies. They're not really 3D strategy that you could do. In 3D strategies, I think if you insert other material, nanoparticle material inside to incorporate that within the cell, you probably will need to still have some way of tensioning, what you put in there in order to have the amount of tension that you need for the directionality. I would say, some of the work from Godana, Volchek, Novakovic, and their Nature paper on how to, exert that electrical stimulation and the mechanical tension is likely to really be what you need in order to have that organization and pattern, and beyond just substrate adding with various material.
Okay, that makes sense. So we have another question from Michael. I wonder if printing your myocytes on a microsubstrate that has appropriately planned origami folds would help with the alignment of desired forces.
Yeah. That is definitely a great idea. We have not tried to do these origami fold and then try to create, tissues with these origami fold. Um, i- um, if I can, maybe there's a creative way to origami fold 3D versions of, the origami rather than origami folding and patterning just in 2D. But yeah, I mean, definitely a fantastic idea of, sort of exploring more in how to, print and fold to create something that's, aligned and tensioned. Um, you know, just a lot of interesting approaches we haven't, had enough time and effort to get into so far.
I agree, and Mike, thanks for sharing your excellent ideas with us.
Mm-hmm.
I think
Mm-hmm.
I encourage you to contact Professor Wu in person, and then maybe look into potential collaborations.
Mm-hmm. Yeah, love to. That's great.
Awesome. So the next question is my next question is about the challenge of having a lot of cells, right? I mean, for your like clinically relevant engineered tissues. So beside the method that you described,
Mm-hmm.
can you comment on the potential application of using bioreactors
Mm-hmm.
to increase the lot size?
Mm-hmm. Yeah. So there's been a lot of interest from companies to create bioreactors to grow cells, especially, say, at least one version of it from Terumo that I've seen involves these little fibers as extra surface area. So you have a drum and it has millions of these fibers, and the fiber increases the surface area, rather than just the monolayer of surface. These fiber each comes with the entire sort of circular sphere, circularized surface area in each fiber that you can grow like a gazillion of them in this drum, and the media fluid to circulate through. I think that's sort of the things that people have been interested in, using a bioreactor to grow a lot of cells. I would probably say that that strategy works quite well for most cells, that it does not, require this, cell contact, phenomenon.
Mm.
Because if you were trying to grow the cardiomyocytes and you cannot passage them, you could passage the drum. It just, takes a lot of effort. That's probably, something that you could do if you try to generate a lot of cells. We were trying to do this with growing the tissue. Again, as I mentioned, thinking about from a perspective of being a developmental biologist,
I see.
instead of growing all of the cells that you need in the drum and then harvest all the cells with a huge number of cells in the drum, it would be maybe a little bit easier in terms of what you want to do by starting with a small number of cells, but apply the growth strategy of that small number to have it in situ grow into the tissue construct that you would like to have by, co-opting these developmental principle. And so
Mhm.
what at least what we're working on next about this whole cell contact business is to see, can we understand the signaling pathway that the cell contact is exerting so that we can essentially inhibit the contact inhibitory signal. So by inhibiting the contact inhibitory signal, the cells will grow as if they are not in contact with each other, so that we can do the cell growth, with the strategy that we implemented to grow that smaller into bigger tissue of interest, rather than having to grow it in a, drum or a 2D or whatever format first, before then harvest it all to try to assemble it, into a structure. In our minds, I think there's some utility and benefit, based on thinking about heart development, in having the cells to be grown next to each other from the start, so that they're connected better, they're organized, hopefully in a certain arrangement better, so that in the end, what you have is something that's much more, like the real tissue, rather than trying to put all the things together at the end. As I said at the beginning, this brick mortar type of engineering tissue, for me, is somewhat tough, because I think once you get all that cells at their end states and then try to assemble them into one, they may not really assemble into the tissue that you were meant to have them be, because the cells had passed through all the developmental processes where many of the interaction is probably taking place early on. And those interactions are not available anymore when the cells have come to the very end. As opposed to if you start with the immature cell, you coach the cell with a different strategy for it to grow, and be next to each other, like what you normally do during fetal development, and let that tissue grow into the size that you want the eventual size to be. To me, that all of those interaction as they develop, next to each other, potentially provides more of a benefit than trying to do this after making all the cells, outside, and then try to assemble it at the end. Yeah.
Wonderful. Thank you very much for your comprehensive answer to my question. So I don't have any other questions. Human, do you have any questions?
Yes, thank you so much for your great presentation. I do have one question that I usually receive once we actually talk about disease modeling and heart-on-chip platforms. People recently asked about integration of the immune system and the systemic interaction with the. I saw that you have the systemic interaction between liver, heart, and other things that you tested. But I want to know your perspective about how to really answer these questions from people, like once we submit grant or in the papers and we talk about heart on chip, and they want us to integrate immune system and systemic interaction in our model.
Yeah, no, that's definitely a great question. How to deal with this multicellular nature of biological interaction in real world when you're doing things in the microchip, format. I think probably, at least in the big picture kind of way, is we need to realize models are never perfect, and they're some models are more useful for some things than others. So what we try to do with the chip format, we have to recognize that certain things we are recapitulating, but we're not doing everything. So I would say certain kinds of multicellular interaction may be easier, like endothelial and cardiomyocytes or fibroblasts, and like those things that make sense, that are juxtaposed within the tissue itself, are the easiest, to have that recapitulated. Once you start going further beyond, when people talk about, immune circulating things or brain-heart interactions, I would say, then you may need to start asking a different question, which is this really the best model, for answering the question? Because once you really get that far, I'm not sure, you should keep staying within that same model system. There may be a different model for trying to address your question is a better one to address than staying with the same, in microchip. So, I would, have to acknowledge I may not be providing exactly what the answer that you might be thinking, like, how do I put that immune cell into my microchip so that it could actually mimic, the immune cell interaction? Again, as I said, I think probably there are a few scenarios where introducing immune cell into this biochip circuit and let it flow and cycle around, it might give you the representation. But my sense is that the further you are from these immediate tissue cell to cell contacting context the further you are from what's really going on in vivo. So I would just, always want to be careful before, just kind of throwing stuff into the system, expecting it to just function like what you think it ought to function.
Fantastic, inspiring. Thank you. Mohsen, I pass to you. There are some questions in the Q&A box.
Yeah, well, thank you very much. So the next question is from Ali from the audience. So thank you for your great talk. I like your term development nearing, and was wondering how far we are from developing mature organs in the presence of limitations such as inappropriate engraftments and the difficulty to fully control stem cell fate. So that's the first part of his question. The second part is, what is your opinion on using decellularized matrices to build functionality and structurally mature organs?
Yeah, those are two fantastic questions, and I think they definitely have been on the forefront of people's mind, in this tissue engineering, regenerative medicine world. So addressing the first one, I would probably say, other than cell of the hematopoietic system, there are very few successes so far in transplant regenerative therapy of any other organ within the body. The sort of next closest, maybe there have been some dopaminergic neuron transplant that was tried in people, variably successful. And more recently, say, corneal transplant of, the stem cell derived cornea, which different, labs in the world are trying, in Japan and some in the US. Those, I think, other than, again, maybe I'll just say that the skin grafting is probably one of the furthest, in terms of therapeutic. And so blood cell and skin cell, generally easier to engraft into people, have been the more successful. Almost anything else, especially ones that require a full 3D solid organ type of functioning, we have not been particularly, so great at figuring out. And I think some of the, there are many different reasons, and I'm sure every organ has its own reason of why it hasn't been nearly, as successful. Like, for example, when people try to do islet cell transplant into liver, there are some sort of further successes. It hasn't been nearly as universally adopted, but I do think that people, who have been doing these cadaveric islet transplant in liver have, been more enthusiastic about the success of doing, that. But, again, liver environment, and a cell like islet perhaps are more easily engraftable in their sort of interaction. For organs, let's say if we start talking about, heart, or if we're talking about kidney, there's just not really been anything so far that people had successfully create and graft. And I think, this comes with the need to understand what kind of function that you're trying to recapitulate, for one, and then also what kind of tissue arrangement and organization, that you need, than the other. So I would probably say that if we can figure out, hopefully by understanding some of the developmental principle of cell patterning, arrangement, and organization, so that we can coach the starting cell into the right arrangement that they need to be, then perhaps we would have something that has more of a chance to become a regenerative organ or tissue for, transplant therapy in the future. But if we keep trying to do cell injections into something that's meant to function like organs, I'm pretty sure we're never going to get anywhere before people eventually give up on the idea of injecting just plain single particle cells, even if combination of different kinds of particle cells, because it's, that's just not how the real final functional tissue, knows what to do with, especially when injected into a hostile environment. So that's sort of my thoughts on the first question. So the second question, the decellularized matrix part. So we've, worked with Harold Ott, Harold Ott, who did a lot of this initial heart decellularization working with Doris Taylor. I would say that the material itself certainly has a lot of the components that the heart is used to because they come straight from a heart that previously was living within those components. So the cells certainly have been able, and this is work that Vahi was doing when he was in my lab, to incorporate the decellularized material. But what we have not seen, and that was early on one of the hope that these decellularized matrix is supposed to do, is this addressability part, meaning cardiomyocyte falls into the cardiomyocyte spot, endothelial cell falls into endothelial cell spot, and fibroblast falls into fibroblast spot. I would say that we just did not see any of those addressability aspect. If you put cells onto this decellularized matrix, it just finds whatever surface that it can find, and sit on it. So again, I think it probably is somewhat like any other matrices that you're using, in terms of what the cells would do in the decellularized matrix. It will land, it would stick, but it won't really organize with decellularized. I think the effort of trying to inject cells into the fully scaffold decellularized matrix is still continuing. Many people are still trying, pretty hard to figure out how to make that work. So, hopefully that they'll find more successes, in being able to do that. But again, I think it's been a little while, and that I suspect that some of the sort of challenges in cell number, that's one thing I think has been a tough, at least from when I previously worked with Harold Ott, and the other is just sort of making sure that you get enough cells that are surviving, with perfusion and localizing in the right place for the decellularized matrix. So hopefully that answers your question on those two things.
Wonderful. Thank you. So I hope you receive your answer, Ali. So Michael asks about your email address, and he wanted to follow up with you. So I guess your email address is available on your website.
Yeah, so it's very easy to just search up, but I could just say that, it's SMWU, so smwu@stanford.edu. Pretty simple.
edu. So I'm just typing it in the chat box, smwu@stanford.edu.
Yeah.
So feel free to contact. Yeah. Professor Wu, if you have any questions or you want to have a follow-up conversation with him. So I think with that, I just wanted to make another announcement that Professor Wu accepted to record this talk, and it will be posted on our YouTube channel. Please follow us on LinkedIn and Twitter, so we can so you can know when the video is ready for on YouTube, on our YouTube channel. Also, I wanted to remind everyone that our next speaker is Professor Todd Orr from McMaster University. He will be talking about hydrogels for tissue engineering and drug delivery applications. Last but not least, I would like to thank our speaker for his excellent talk, very inspiring and very informative talk. I really enjoyed your talk, and I'm sure that Human and then the participants also enjoyed your talk very much. And also I wanted to thank our participants for joining this event. With that, I'd like to end this session and then wish you all a great day or night ahead of you. Thank you very much.
Thank you, Professor.
Thank you so much. Yep. Thank you so much for doing that. Take care.
Bye-bye.
All right. Thanks.
Thank you.
Bye-bye.